DNA replication is initiated in eukaryotes in two distinct phases: origin licensing and origin firing. Origin licensing occurs in late mitosis or early G1 phase of the cell cycle. In this phase, the pre-replication complex is formed, which ultimately leads to the assembly of the MCM27 complex at the replication origin in an inactive double hexamer. The second phase, origin firing, occurs during the S phase of the cell cycle. During origin firing, the MCM27 complex is converted to an active replication fork helicase and DNA synthesis begins. Origin selection and licensing. So what actually determines the origin of replication? This depends somewhat on the actual type of eukaryote. First, let's look at a budding yeast, such as Saccharomyces cerevisiae. In these simpler eukaryotes, origin sequences are characterized by autonomously replicating sequences, or ARS. These DNA sequences are 100 to 200 base pairs long and are present on all chromosomes at roughly 30 kilobase intervals. Therefore, there are around 200 ARSs in a typical budding yeast genome. A consensus ARS sequence alone is not sufficient to define an origin. However, mutations in these cis-acting sequences have been shown to alter origin efficiency. The other contributing factors in defining a replication origin are discussed later. Within an ARS, there are cis-acting DNA elements called the A, B1, B2, and B3 elements. The A element is highly conserved across lower eukaryotes. The conservation of the A element allows for an extended ARS consensus sequence, EACS, of 17 AT-rich base pairs. The B1 element provides an additional sequence for recognition by the origin of replication complex, ORC. All three B elements are thought to help facilitate helicase loading and DNA unwinding. Local chromatin structure also plays a key role in origin efficiency and firing timing, and does so independently of the specific origin sequence. The BA-H domain of the ORC1 subunit plays an important role in origin selection as it interacts with chromatin in all eukaryotes. Studies have shown that in metazoans, the ORC1 BA-H domain binds the histone methylation H3K20Me2. This histone modification is catalyzed by the methyltransferases SUV4-20H1 and SUV4-20H2, and is the most abundant histone modification. Thus, it is clear that this modification is not a means by which the ORC is directed to specific loci, but likely serves to stabilize the ORC complex via ORC1. Another histone methylation, H4K20Me1, is catalyzed by PRSET7 and has been shown to be critical for pre-replication complex assembly. Almost all the origin sequences in S. cerevisiae maintain a nucleosome-free region at the ACS. This NFR is determined by the origin sequence itself and is present whether or not the ORC is bound. The absence of nucleosomes provides a sort of landing pad for the ORC. In yeast, the vast majority of replication origins are flanked by precisely positioned nucleosomes. Displacement of these nucleosomes has shown to impair helicase loading and pre-replication complex assembly. Due to this, it has been hypothesized that these nucleosomes may interact with MCM27, thus providing stability and facilitation of helicase loading. Budding yeast are one of the only eukaryotic organisms which harbor consensus sequences at their replication origins. Metazoans lack any apparent consensus origin sequence. In fact, in the rapid early cleavage stages of Drosophila, replication initiates at random sites with regards to sequence. In higher eukaryotes, several genetic features, both sequence and chromatin dependent, are associated with origin activity. These include chromatin features such as DNase-1 hypersensitivity sites, and primary sequence features such as those that can form G4 quadruplexes. The degree to which these features influence the determination of origin sites is unclear, however, due to the limitations of genomic techniques, particularly when studying metazoan genomes. This is complicated by the fact that origin usage is heterogeneous across cells and metazoans. Origin activation and timing. Replication timing patterns are far more conserved across eukaryotic evolution than specific origin positioning. Origin firing occurs by the maturation of licensed origins from the pre-replication complex to the pre-initiation complex. Not all origins which are licensed will end up initiating replication. Generally, early replicating domains are euchromatin regions located in the inner nucleus. They are characterized by high gene density and high G and C base pair content. Conversely, late replicating origins are characterized by fewer genes and are generally packaged in heterochromatin near the nuclear periphery. Normally, centromere DNA is early replicating and influences the replication timing of nearby origins. The mechanism behind this is that kinetochores attract the DDK enzyme, promoting early replication. Subtelomeric regions replicate late, the timing of which is influenced by local chromatin structure. Origin firing of these subtelomeric regions is delayed to late in S phase by histone deacetylase SIR2 or by the Ku complex. Acetylation of histones H3 and H4 has been shown to be important for efficient origin activation and DNA replication in S phase. Among the key determinants of origin firing timing in yeast are the transcription factors FKH1 and FKH2. These proteins promote early firing in some origins through ORC binding and association with CDC45 during pre-replication complex maturation. FKH1 and 2 binding sites are limited to early origins, however early firing is also dependent on close proximity to the ACS. The telomere binding protein, RIF1, is also an origin timing regulator which is conserved from yeast to humans. In its absence, there is a premature activation of normally late firing origins near the telomeres and subsequently earlier replication. Studies in fission yeast have shown that RIF1, along with another protein, TAS1, regulates the timing of CDC45 SLD3 loading onto the MCM27 complex. As will be covered later, this is a key step in origin activation and believed to be a rate-limiting step controlling replication timing. In S. cerevisiae, RIF1 contains two GLC7 or protein phosphatase 1 interaction motifs. The phosphatase opposes DDK phosphorylation of MCM4, thereby repressing origin firing. This is overcome by rising DDK concentration late in S phase, as DDK also phosphorylates RIF1, which releases the phosphatase subunit. Local chromatin structure not only affects origin selection and licensing, but also influences origin timing. Nucleosome positioning, which is established during G1, differs between early and late origins, and is modulated during origin activation in the cell cycle. Chromatin structure may also influence origin activity due to accessibility of initiation factors. As discussed earlier, the local chromatin environment regulates ORC recruitment and pre-replication complex assembly. The timing of ORC binding and pre-replication complex formation during mitosis and G1 phase has been shown to influence origin efficiency and firing timing during S phase. Lastly, origin activation may be prevented under replication stress or DNA damage by the S phase checkpoint. This is mediated by the phosphorylation of DBF4 and SLD3, as well as the inhibition of CDC45 recruitment. Although there is strong evidence in support of epigenetic control of replication timing in metazoans, there is also compelling evidence for the role of primary DNA sequence. Experiments in transgenic mice indicated that the locus control region controlling expression of the beta-globin gene was able to dictate local replication timing in a transcription-independent manner. In humans, the beta-globin locus is duplicated early in S phase in erythrocytes, but late in other cell types. Furthermore, targeting histone acetylases and deacetylases near a mapped initiation site at the human beta-globin locus was shown to accelerate or delay respectively replication timing of the local region.